These MCQs are for the NEET examination. The chapter is Biotechnology Principles and Processes. All these questions are based on the NCERT book. First question is, which one is a plasmid? See, in the four options, correct answer is B, the PBR 322. So this is a plasmid. The remaining three are the restriction enzymes. Next question is, which technique is used for separation of DNA fragments? See, the gel electrophoresis is used for the separation of DNA fragments. Therefore, the correct option is A. Next is identify the wrong statement. We have been given the four statements and we have to identify the wrong statement. The first is plasmids and bacteriophages are used as cloning vectors. Correct. Genes encoding resistance to antibiotics are used as selectable markers. Yes, this is also correct. Next is selectable markers help to separate transformants from non-transformants. Yes, this is absolutely correct. Next statement is Agrobacterium tumefaciens can deliver a piece of DNA in animal cell. This one is wrong because Agrobacterium tumefaciens is used to transfer the DNA in dicot plants and not in the animal cell. So our correct option is D. Align DNA can be transformed into host cell with the help of. So next question is, alien DNA can be introduced into host cell with the help of microinjection, ballistic or gene gun method and use of disarmed pathogen. So all these tools are used for transfer of DNA into host cell. So therefore the correct option is D, all of these, because all of these methods can be used, or all of these methods are used for transfer of DNA into the host cell. Next is treatment with which cation increases the efficiency with which DNA can enter the host cell. See, the calcium cation is used to increase the efficiency of the host cell to uptake the DNA. So correct option is A. Next is source of Taq polymerase used in PCR is. So in the PCR reaction, the Taq polymerase enzyme that is which is used. So this is actually the source of this enzyme is Thermus aquaticus. Next is which of the following is not a palindromic sequence? So palindromic sequence is that which is same when it is read from the 5 prime to 3 prime and or the 3 prime to 3 or the 5 prime in both the stands. In the four options, the option D is not a palindromic sequence because so this is not the same in both the strands. Next is assertion and reasoning type questions. So we have been given that we have been given the two statements. One is assertion and second one is reason. And for selecting the correct answer, and we will mark it A. So our option will be A if both assertion and reason are true, and the reason is a correct explanation of assertion. And we will mark it a B if the both assertion and reason are true but reason is not a correct explanation of A. We will our correct option our will be C if assertion A is true and reason R is false, and our option will be D if assertion is false and reason is true. Now let's see this assertion and reason type of question. Uh, Assertion is that DNA cannot pass through the cell membrane. The reason given is that DNA is a hydrophilic molecule. See, the assertion is also right. The reason is also correct. So the reason are the DNA is a hydrophilic molecule. This is a correct explanation of assertion that DNA cannot pass through the cell membrane. So DNA, due to being hydrophilic molecule, it cannot pass through the cell membrane. So therefore, our both are correct, and reason R is a correct explanation of assertion A. So therefore, our option will be A. So correct option will be A. Next is assertion reasoning type question. Next is assertion is in gel electrophoresis DNA moves toward anode. The reason given is DNA is a negatively charged molecule. The reason R is a correct explanation of A. Therefore, our answer will be A. Next is molecular scissor used for cutting DNA is. So see, the restriction enzymes are also called as a molecular scissors and they are used for cutting DNA. So correct option is D. Next is polymerase chain reaction is used to synthesize. See, the polymerase chain reaction is used to synthesize multiple copies of genes. So therefore, our correct option is A, multiple copies of genes, and it is not the proteins or RNA. So this is used only for the multiple copies of genes. Okay, the thank you.